Welcome to the BioInsights, where science comes alive.
Today, we are going to explore what is quite literally one of the most powerful separation techniques in all of modern science, chromatography. Think of it kind of like the ultimate biological sorting machine. It's a tool that absolutely revolutionized how we understand and manipulate the very building blocks of life. But let's set the stage, right? Imagine the immense, almost impossible challenge scientists face every single day in the lab. Picture a cellular lysate. It's this chaotic, microscopic soup containing thousands of different proteins, nucleic acids, and just random cellular debris. Your job is to extract one single, tiny target molecule from that crazy mixture with absolute precision, and you have to leave everything else behind. I mean, how is that even physically possible?
Well, here's our roadmap for figuring that out. We're going to define the protein purification problem, walk through the 4 universal steps to solve it, and then break down the 3 big techniques: affinity, ion exchange, and size exclusion, before looking at some real-world biomedical impacts.
All right, let's start right at the beginning with Section 1, the protein purification problem. You know the old saying about trying to find a needle in a haystack? Well, imagine if that haystack was actually full of pins, nails, and wire. That is exactly what scientists are up against. It's not just that the target is microscopic, it's that your one specific needle is surrounded by thousands of other biomolecules that share almost identical physical and chemical properties. So, to pull our specific needle out, we literally have to exploit incredibly subtle, unique traits of our target molecule.
Moving on to Section 2, let's look at the 4 universal steps used to tackle this.
The cool thing is, virtually all chromatography relies on a standardized 4-step workflow. First up, you equilibrate. This just means prepping your stationary column with a specific buffer to get the exact right pH and ionic strength. Second, you load your sample. This is the magic moment where your target binds to the column matrix. Third, you wash it. Basically, you're flushing out all that non-specific, unbound cellular junk that we don't care about. And fourth, you elute. You purposely change the chemical conditions to break the bond, releasing your highly purified target right into a collection tube. Let's dive into our first specific technique in Section 3, affinity, or the perfect match. Now, this is arguably the sharpest tool in a biochemist's shed because it relies entirely on biological recognition. And to really grasp this, we've got to talk about the ligand. A ligand is immobilized on a solid support matrix, and it acts as a highly selective lock for a very specific target key. It will literally ignore everything in that soup except its exact biological partner. We're basically borrowing nature's own flawless targeting systems, like an antibody grabbing an antigen and putting them to work inside a glass column.
But here's the catch. Sometimes a target's binding site is buried super deep within its structure. If you just glue a ligand directly to the column matrix, your target might just clunk against the wall, a problem called steric hindrance. So, to fix this, we use these things called spacer arms. It's just a chemical chain that pushes the ligand out into the fluid so it can actually reach into the target. A really fantastic example of this in action is immobilized metal affinity chromatography, or IMAC. Scientists can actually genetically attach a little His tag to a protein, which then binds incredibly tightly to a nickel-based ligand. To finally elute it, you just toss in a competitor, like imidazole, which kicks your purified protein right off the matrix.
Okay, on to Section 4, ion exchange, or catching charges. This concept here is absolutely foundational. As you probably know, proteins are made of amino acids, right? Well, their net electrical charge actually changes depending on the pH of the liquid they're floating in. The isoelectric point, or pI, is the exact pH where a protein is perfectly neutral. If your buffer's pH drops below this point, the protein gains protons and becomes positively charged. If it goes above it, the protein becomes negatively charged. And this brings us to that classic rule, opposites attract. This is the heart of ion exchange. If you tweak your buffer's pH so your target protein goes negative, you're going to run it through an anion exchanger, which is just a column packed with positively charged resin beads. On the flip side, if your target is positively charged, you use a cation exchanger packed with negatively charged beads. Your target sticks to the opposite charge, while all the junk you don't want just flows right out the bottom. So, how do we finally get our protein back? We have to break those electrical bonds. And there are 2 brilliant ways to do this. Strategy 1, you can slowly increase the ionic strength by adding a salt gradient. The salt ions literally compete with the protein for those charged spots on the beads, eventually knocking the protein off. The proteins with the weakest charge fall off first. Strategy 2 is even simpler. Just alter the pH past the protein's isoelectric point until it totally loses its charge and lets go of the column. All right, moving to section 5, size exclusion, the maze. Okay, get ready for a totally counterintuitive rule. In size exclusion chromatography, sometimes called gel filtration, the largest items actually win the race. They exit the column first, while the smallest items exit last. No way, right? It seems totally backwards until you actually look at the structure of the column itself. Think of an ant and a beetle trying to travel through a field of tall grass. Our column is packed with spherical gel beads that are completely full of tiny pores, like microscopic sponges. When you pour your sample in, the large molecules, our beetles, are physically just too big to fit into those pores, so they bypass the beads entirely and wash straight down the empty space, the void volume, exiting super fast. But the small molecules, our ants, wander into this incredibly intricate maze of pores inside every single bead. Because they're taking this massive detour, they are delayed and elute much later. This trick perfectly separates a sample strictly by decreasing molecular weight.
Finally, section 6, real-world biomedical impact. So, we've talked about the mechanics, the biological locks, the electrical charges, the porous mazes. But why does mastering this molecular sorting actually matter? Honestly, without chromatography, modern medicine just wouldn't exist as we know it. It is the absolute gold standard for protein purification. We use these techniques to isolate biomarkers from blood for early disease detection. Pharma companies use massive columns to manufacture incredibly pure therapeutics and vaccines. Even size exclusion plays a huge everyday role. It's used constantly for desalting samples, quickly separating big therapeutic proteins from tiny, potentially harmful salt ions. To wrap things up, here is a quick cheat sheet for how we isolate biomolecules. We use affinity to sort by biological fit, leveraging those precise lock-and-key ligands. We use ion exchange to sort by electrical charge, pulling molecules using pH and salt gradients. And we use size exclusion to sort by physical size, sending them through a maze of porous gel beads. And in the real world, scientists will actually chain these techniques together to get thousands-fold purification in just a few steps.
It's pretty mind-blowing when you think about it. We are no longer just relying on natural biological interactions. We're out there actively engineering incredibly stable synthetic biomimetic ligands right in the lab. As our ability to separate and purify the building blocks of life becomes sharper and more sophisticated, I really hope this explainer leaves you wondering, what completely new frontiers in personalized medicine is chromatography going to unlock next? Thanks for learning with me today, and keep exploring the microscopic world.